Okay. All right. Well, thank you, Alan. Yeah, thank you all for coming and listening to me, and thanks, Alan, for the opportunity. I'm always excited to get to share stuff, um, so what I'm going to talk with you guys about here. Wait, let me see if I can get this to work. There we go. So first of all, I'm going to start off just telling you a little bit about my story of how I came into statistics and how I ended up where I am today, and hope that can be something that is interesting and informative to you guys. And then I'm going to talk a little bit about what my life is like as a biostatistician at MD Anderson. I'll also talk about opportunities, including grad school and job opportunities at MD Anderson, to let you guys know about those. And then for the rest of my time, I'm going to try to convey to you what I see as some of the deeper impact that we statisticians can have in the scientific world. Many times, where people think of us as technicians, where I'm going to try and make the argument that we're scientists and we can actually make deep impacts in many different ways. So I'm going to hit a couple different areas where we can do that. One is in new methods. I'll talk a little bit about big data that I've worked with and has really kind of proliferated all around scientific areas. And I'm going to talk about experimental design and talk about a case study that shows how important that is. And then I'm also going to talk a little bit about how we can help refine thinking and even help lead the science. And I'm going to talk about some exciting stuff that we're doing in colon cancer here at MD Anderson that I'm having the opportunity to participate and serve a major role in. That's really enjoyable. And then finish with some conclusions. So first of all, I know you all are statistics majors, so you know what statistics is. But what I want to hit on here is get you to see that statistics is actually a scientific field. So this is just the Webster Dictionary of statistics. Basically anything involving collecting, analyzing, and interpreting data. But rather than, again, thinking of us as just technicians, really you could think of statistics as the science of data. It's the science of how to collect data and be able to make inference on populations through proper analyses. So it's actually a scientific field, and given how across society data is central to almost every venture, it really is a field that is placed at the center of a lot of the most important questions in society. And so society has kind of recognized that, where you probably hear the word data science thrown around, and it kind of means somewhat different things to different people. But what I would argue is that statisticians are the original data scientists. I think data science in general includes a lot of other quantitative scientists that might not be statisticians, but then within that broader field of quantitative scientists, I think where we fall as statisticians is we're the ones that best understand variability and uncertainty. So I think we need to remember that so we can really make the biggest impact. But statistical ideas are the key to any enterprise in which data are collected and conclusions are drawn. Okay, so for me, how did I end up in this field? Well, I did my undergraduate degree at a small liberal arts school called Messiah College near Harrisburg, Pennsylvania. And I was a math education major at the time. And then there was Dr. Marlon Eby, a statistics professor in the math department, who started a statistics minor. And he would take it upon himself to identify math students and encourage them to consider statistics. And he would have people come in and speak about opportunities in statistics, kind of like I'm doing, I guess, to your class. And so I saw the opportunities there. And the other thing he did was encourage me that he thought I had the ability to succeed and go to graduate school. And that really opened my eyes, because I was only the second person in my family, either side, to even complete an undergraduate degree. I had an uncle a couple years ahead of me who did. So the idea of even graduate school, much less a PhD, was not even in my mind. But he encouraged me that he thought I had the ability to succeed there. So that opened my eyes to opportunities. And so what I really liked about the field was that this was an area where I could use my math skills, but to solve real world problems. And I've always enjoyed learning new things. And I forget if it was George Box or some famous statistician said, the great thing about statistics is we get to play in everybody else's sandbox. And that really appealed to me, to get to always be learning about these different areas and contributing in different areas. So I did finish my math education major and took a little break and went and taught high school 2 years and did some coaching, and then went back to graduate school there at Texas A&M in statistics. And my original thought was, I thought maybe I'd want to be an undergraduate teaching professor like my mentor Marlon Eby, And I thought, or I thought maybe I'd get a master's and then get a job in industry, because I know there's a lot of good jobs with a master's. So that was kind of my initial thought. So really, a PhD wasn't really something I even at that time consciously thought about. But then after my first year, I passed the qualifiers at the PhD level, and my professors, many of whom you still have there, were some of my big encouragers. Sujin Wang, Dr. Longnecker, and others that were my first year professors really helped me out, encouraged me to stay for PhD. So then I decided to try and stay for PhD, and I did an internship at a pharmaceutical company, and I kind of saw how the opportunities can increase for you the further you go. So that's one thing I want to encourage all of you to keep in mind, is that so I think the bachelor's program, I'm really excited that's going there at A&M. I think that's a great thing, and I know that there's jobs you can get with a bachelor's, but I think if you go get a master's, you'll have even more opportunities at that point. And if you go get a PhD, then it also increases the opportunities that you have. And what I found was, at the time when I was going, there wasn't really much of an undergraduate degree, but I was deciding between master's and PhD. And one thing I saw is that if you get a PhD, you tend to have more opportunities to sort of be in leadership, where with a master's, you might end up having to sort of follow other people who have PhDs, even if you might know more about what's going on than them. So the degree sort of gives you more opportunities. So one thing I encourage all of you to do is, keep working hard, studying this field, but really take seriously the feedback of your mentors. And even if you don't think that you could do graduate school, or a PhD or a master's, or you don't think you would want to take that time, I encourage you that time goes faster than you think. And also, don't limit yourself by what you think you can't do. Because I know if I did that, I would have never tried most of the things I've tried and been able to do. I never knew I was capable of them before. So that's kind of my word of encouragement to you. But then, so while I was a graduate student, then I got involved in some interesting cancer research projects with Professor Carroll. And he has an ongoing collaboration with the nutrition group there at Texas A&M, and they were doing research looking at how different nutrition factors affected the risk of colon cancer. And I got very interested in the science of what they were doing, and also the data they were collecting was kind of intricate. So I realized that I really liked collaborative research, working with scientists, and I enjoyed in particular working on cancer studies and learning about cancer and contributing to understanding cancer. And also I realized I enjoyed methodological research, being able to develop new methods, especially for complex data. And so when I finished up, so honestly, I never thought about becoming a research faculty in statistics, honestly, until right at the end of my PhD. Then when I realized how much I liked methods research and collaborative research, and I realized that being a tenure track faculty member is when I could really do that the best, I sort of swallowed my fear of failure and my own doubts and chose to believe what my mentors encouraged me, that they thought I could do it. And I thought, and if I could do it, I thought I would like it. So I gave it a shot. And so I ended up coming here to MD Anderson as an assistant professor. And so here at MD Anderson, then, there's really two parts to my job, two main parts. One is collaborating with biologists and medical researchers on cancer research problems that they're working on. And that takes many forms. A lot of it is clinical trials where they're treating patients and testing new cancer treatments, but it also includes laboratory studies where they're trying to identify potentially new drugs, new biomarkers that they can use to help treatment, and to learn the basic biology of the cancer that could lead to more new drugs and new biomarkers. And so that involves helping design studies to make sure they can answer their chosen hypotheses that they have, and also to analyze the data, make sure they draw proper conclusions. So then I'll help them write papers, help them submit grants for research funding, and make sure that their plans are statistically sound. So then the other half of what I do, and really my position here really is like 50-50 in these two areas, is to develop new methods. And so something I don't do too much of, but some of my colleagues do a lot of, is new clinical trial designs that are more efficient and can learn quicker whether a drug is working. And that's a big area where I think our department has made a lot of impacts in this through the chair when I was hired, Don Barry, is a world leader in this area and has really helped change the way things are done. So there's new designs. And then where I've worked more is methods to analyze complex data that emerges from some of these new technologies. And I'll talk a little bit about that later. And so again, as a practical matter, I write papers on these, develop software, write books, book chapters. And one great thing about being in academia and doing research is you get to travel a lot, give talks, meet all kinds of interesting people all over the world, and of course, get grant funding to help fund these things. So that's kind of the, what I do here. So just as sort of, big picture of what are opportunities for statisticians here at MD Anderson, and I think in general at a lot of places like this, there's faculty. We have tenure-track faculty and tenured faculty like me, where we do methods and collaborative research, and then sometimes there's also research faculty that focus mostly on the collaborative side, not so much on new methodology, that those require PhDs. We also hire many statistical analysts. We have about 40 of them that have typically master's degrees in statistics. Some of them go on and get their PhD, even as they're a stat analyst. And so they're working primarily on the collaborative stuff. And so we kind of work as a team. And so with having so many stat analysts working that way, we don't have to get caught up in doing all the routine analyses as faculty. That's why we have the time to do some of the bigger picture stuff in research that we do. And then we also have postdoctoral researchers, and we have graduate students. And so we have really two main graduate programs, and kind of a third because we have a connection with you guys there at Texas A&M. But one we have is Graduate School of Biomedical Sciences. That's part of the University of Texas system. We have our own PhD in biostatistics, and then there's also another program in bioinformatics that we admit students into. Then we also have a joint biostatistics PhD program with Rice University, and we have a training grant that helps fund that from the NIH. That's another alternative. So then those students get Rice degrees, but we can also, either Rice or MD Anderson faculty can serve as the advisors. And I'm the MD Anderson side director of that program, so I'm heavily involved in administering that as well. We also have a contract with your department at Texas A&M where we can work with Texas A&M students, and that gives Texas A&M graduate students an opportunity to get involved in cancer research and work with some of us as well. And there's one student, Elizabeth Jennings, now McGuffey, who was Ray Carroll's student that my colleague Vera and I co-advised with him, and she worked with us on things. Now she has a faculty appointment at the Naval Academy. So there's a lot of opportunities. And so for you guys in the undergraduate program there, if you want to go to graduate school, I'd encourage you to take a look at our graduate programs, as well as the excellent program you have up there as well. Okay, so one thing I want to say is if any of you have questions for me as we go, feel free to ask questions, and I'll just keep going if you don't ask, but don't feel bad about asking questions if you have any. I'll try and leave time at the end for questions as well.
We do have a question.
Okay. Yes.
I'll probably have to restate it, but we'll let him ask real quick. Go ahead.
So before you switched to biomedical field, did you have any background before that? Like, did you know anything before biomedical, before you switched to biomedical?
Okay. So the question was, before you became involved in biomedical research, what kind of background in biological sciences did you have? I think where that's coming from is, in order to do this type of stuff, do I have to have a degree in biology and know a bunch of genetics from the get-go?
Right. Well, one thing I'd say is it's great to get it, if you want to go in that area, get as much background as you can. But the funny thing is, for me, I had almost none. Because like I said, I loved learning, and that was true. But the one thing I hated was biology. It was the one thing I hated in high school. In undergrad, I avoided it because I was bored with just memorizing all these big terms that didn't mean anything to me. So I really had almost none coming into graduate school. And then because I worked with the nutrition group there, I would go to their lab meetings, and I learned just sort of some of their biology there. And that's when I became interested, because I realized that biology, especially when you're doing research, is like a really complicated puzzle that's really interesting, actually. And it's not just about a bunch of long, meaningless terms. So then I got to really enjoy biology. And so I had a lot of catching up to do sort of to learn some background, but I've sort of learned it as I've gone. So even when I was hired at MD Anderson, I knew not much at all, but I've learned a lot on the job. And so I think you can learn a lot, just given the good scientific and statistical skills that you can develop as a statistician. You can go into a lot of fields and learn enough of the science to be relevant, but you have to really talk with the collaborators and really try and think about it and understand it. Because one thing I say is statisticians who don't try to learn the science and they just try and stay super shallow, I think have a more limited impact. Where if you just try and learn the key ideas of what they're doing and try and communicate with them, it takes some effort, but then you can be a lot more effective in what you do. But one thing I think that's changed, so I was a grad student basically 20 years ago, and now I think there is more opportunities to get biology background as part of like a graduate program, for example. So like our GSBS and Rice programs, there's some 1 or 2 biology courses you'll take that give you kind of an overview. And I think that's a good thing, definitely, to do. But sort of to answer, part of your question of if you don't have a background in biology, can you not go into biostatistics? That's not true. You can pick it up as you go.
Thanks.
Any other questions?
Yeah, we got at least one more. Go ahead.
So he's been talking about all these graduate opportunities, but if we're just doing our undergrad as of right now, is there anything or opportunities that he knows of to kind of get our foot in the door to even see if that's what we want to do?
Okay. Question is, did you hear that?
I could hear that pretty well.
Okay.
So you're saying, are you talking about MD Anderson in particular or in general?
Just in general.
In general. I think there are. I haven't explored a lot of that myself. Like probably Dr. Dabney can tell you more about some of those opportunities. But I know I've heard of many people getting hired, especially with companies. Many companies hiring people with undergraduates degrees in statistics. But I haven't pursued those myself, so I don't know the details of it. But I know I've heard people talk about that. But I think the general principle is that with more advanced degrees, there'll be more opportunities. But certainly with an undergraduate degree in statistics, you're developing a skill set that is so marketable. I'm confident you can find good jobs out there and do things in different industries.
Yeah. Okay. Thank you. Other questions? All right, please proceed.
All right. Okay, so now I'm going to kind of shift into the other part of my talk, talking more about what I see as, the big picture of what statistics can contribute and showing you some examples to hopefully get you fired up. So a big thing I mentioned is, as a statistician, are we technicians or scientists? And I can say honestly, like one thing that's interesting in working in a medical environment is, working with doctors and stuff, and sometimes like doctors are very busy, so they sometimes interact very quickly and tersely. And sometimes there'll be a doctor that you can tell that the way they want to interact with you very quickly, they're just like, just get me my stinking p-value so I can get out of here. They're not really all wanting to really, ponder and think deeply about what's going on. And actually, I mean, that's not true of most, but sometimes you encounter that, and you can kind of see where there's a subset of people out there that's not small that think of statisticians as just a technician, that we're almost like thinking of us as a machine, that you feed us the data, we give you a p-value to use in your paper. And so sometimes collaborators will interact with you that way. And so when I get that, I don't take it as an insult. I just realize I have to try and help educate them about how much more I can help them with. And many times, collaborators will realize once they work with us what we can offer. So I'm going to try and describe a little more about what I think are some of the deeper things we can offer. But one thing is about collaboration is central to all of statistics, no matter whether you work in industry, government, or academia. Biostatisticians work with other scientists, with policymakers, with people in the company making decisions. So it's an inherently interactive and collaborative field. So one thing is anything you can do to improve your written and verbal communications will help you. But then the nature and extent of the collaborative role might differ depending on the setting. but the principles are the same, that really our value is defined by what we can add in collaboration with other professionals in the discipline we work in. And so what is our role as a statistical collaborator? Well, we do have, obviously we analyze data, we do power calculations, we develop statistical analysis plans, and we do those things, and those are important. But is that all there is? Are we just machines giving them p-values? And we're not. And so getting it back again to that first slide, statistics is the science of data. What we really are is we are scientists who are experts on how to properly design studies and analyze data so that they can draw the conclusions they want to draw. And many scientists do not understand how to do these things properly. And I'll give you one example of when it was done very poorly by some scientists with disastrous results. But you see examples all over the place where people have poor experimental design, improper analysis, they draw wrong conclusions, and take society in a major direction that it shouldn't go. And so we really have a central role there. So our challenge is to help collaborators to see us as collaborating scientists and not just technicians. And so there's different areas where we can make a deeper contribution. So I'm going to hit on 4 of these areas now for the remainder of my time. So first one, methodology, developing appropriate and potentially innovative analysis methods. Okay, so we, they bring us their data. They might expect us to do something kind of basic and simple, but sometimes they have aspects of their data that are really complicated, and the analysis they're asking for might not really get all the information from the data. So that's where we can potentially develop new methods for them that can better get the information out of the data. And that's what, a lot of my methodology research, that's what I'm trying to accomplish there. So I'm going to kind of go off on a tangent and talk a little bit about, what that is about and why it's important in biomedical research. So one thing we see is there is an explosion of big data that tends to be very complex. This data contains lots of information that is really revolutionizing biomedical research, and not even just biomedical research, really all areas of society. And so one area where this is happening in biomedical research is in molecular biology. What is going on inside of the molecules? And in cancer, that's particularly important because cancer is a disease where the typical molecular behavior within our cells gets broken. Because cancer is our own cells that have gone awry. And so what happens is that some of our cells might acquire the ability to ignore the regulatory signals coming from the rest of the body and do what they want. They can multiply at will, refuse to die when it's supposed to be their time to die, and sometimes they even develop the ability to move through the body and generate blood vessels. And I could even add that some cells acquire the ability to become stem cells that can become other types of cells. So all these things are outside of the control of the cells in our body. And honestly, like when you study biology, it's amazing to me just how carefully designed and interactive everything is, and how everything is regulated. It's an incredibly complex system with all these backup systems. That's just amazing. But sometimes things go wrong in the cells, and that's what happens with cancer. And so if we can study what's going on molecularly in the cancer cells, we can understand what's going on in the cancer. And so the field of genomics has really risen in the last 20 years to give us information across the whole genome on many different areas. And so here's just a diagram of some of the different things. We have measurements of the DNA of the cell, the genotypes, copy number, different mutations that go on the DNA. But then the DNA is a template to make messenger RNA, which is a template to make proteins. So we also get direct measurements of the proteins. We get measurements of this messenger RNA to see which genes are being expressed and producing proteins. And then there's these epigenetic processes that are things that regulate and affect the ability of the DNA to get turned into messenger RNA. And so really what's going on in cancer is there's different things in these levels are broken. Then these things then contribute to phenotypes, things we get to observe, and sometimes it affects things we look at as imaging, and all these things affect whether a person has cancer or not, and if they have cancer, all these things affect their survival. So you can see all these different areas. Well, in each of these areas, they've developed assays that can take sometimes hundreds of thousands of measurements looking at the entire genome of what is going on in these cells. And so each of these platforms gets really big, complex data with very complicated issues, and we have to deal with those. Okay, so that's one area of big data. Another area of big data in cancer and other areas is imaging. So there's many different imaging tools that we can use to kind of look at what's going on the inside of someone's body. There's different types of imaging that are good for different things. So CT scans, MRI, PET scans. There's neuroimaging to look at what's going on in your brain and even look at functionally what parts of your brain are active during different tasks, so they can understand cognitive things going on from imaging. And also structural imaging to look at, different structures within your body. So again, this imaging data, very high dimensional, very complex, a lot of information there. So the question is, we know there's a lot of information in these data. How do we get this data out? Or how do we get the information out of these data? Well, I'm gonna tell a little story. You've probably heard the story of the man looking for his keys under the lamppost, but if not, I'll tell the story now because I think it sort of epitomizes the way that people deal with these big data. So there was a man who was down on his hands and knees under a lamppost, and an officer comes over and says, "Oh, excuse me, sir, what are you doing?" He said, "Oh, I'm looking for my keys." He said, "Oh, did you drop them here?" He says, "No, I dropped them over in that dark alley." The policeman's kind of confused. He's like, "Well, why are you looking at them here? Looking for them here?" He said, "Well, the light's better here." Okay, and so I love that story because it characterizes in human nature when we solve complex problems, many times we don't look for the answers where they're most likely to be. We look for the answers where we hope they are, where it would be convenient if the answer was there. And so that type of thinking is kind of dangerous sometimes. And I think that's the kind of thinking that many people use in dealing with complex data. So we have this complex data, and the thought of dealing with this high dimensional complex data, especially to biologists, but to even other scientists, other people in society, is like going down a dark alley. Very scary. Okay, so what do? Well, the typical approach is to simplify the data, to first of all just extract simple summaries from those data, and then analyze those summaries using standard statistical tools. And so this gets rid of the complexity, and this can be a good approach as long as those summaries contain all the information in the data. But the problem is if those summaries that they extract from the data don't contain all of the information in the data, information is lost because they don't look back at the original data anymore. And that is in both of those areas, in multi-platform genomics and in imaging, many of the analysis approaches they use are this approach, which I call feature extraction. They extract some features, throw away the original complex data. So again, the question is that choice driven by the science because they believe those summaries contain all the information, or is it just driven by convenience? Because if it doesn't contain all the information, they have no idea how to deal with that high dimensional complex structured data, so they're just doing it out of wishful thinking. Okay, so that's been a lot of my research program methodologically, is to try and develop flexible methods that can model the entire complex data set as it is, so information is not lost, while accounting for their structure and integrating known scientific information into the modeling. So trying to build tools basically to allow researchers to, instead of just looking under the lampposts, to actually explore the dark alleys where maybe the key information lies. Okay, so again, I didn't go into any specifics, but that's kind of a big picture of motivation. And there's a lot of methods research that you could characterize that's going on right now that's real important that could be characterized this way. Okay, so methods to get information out of the data, that's an important thing. The other thing is proper interpretation of study results. Many times people collect data and then they want to draw interpretations that are really not supported by the data. So here's a nice little comic here, good old Dogbert the CEO. Let's go ahead and read over that. So I kind of like this because it's kind of funny, but the scary thing is in how many areas of science this is effectively what they're doing. They're collecting a lot of data and they're finding something that looks like it's a signal. And they don't ask the question whether it could arise just by chance. They just say, hey, here's a signal. Well, let's interpret it. Okay, and a lot of times it could be something, they could just be looking at a lot of things and picking out the one that does best by chance. So the problem with that is when someone goes to try and reproduce that research, it doesn't get reproduced because it was a chance result, not a true result. So it's important that we make sure that the analysis results are properly interpreted and conveyed. And so a couple errors of thinking that I've seen commonly in my experience. One is proper interpretation of the statistics. Like, I know you guys have, I assume, already talked about hypothesis testing and talked about how you have a null hypothesis and you see whether there's enough evidence to reject the null hypothesis. And then if there's not, you fail to reject the null hypothesis, or you might say you accept the null hypothesis, but you would never say you prove the null hypothesis, at least not if you want to get a good grade in your classes, in your stats classes, right? And it's amazing that I've encountered, one example I have is there's a physician that does many studies in this one area of cancer, and he did all these trials for like 5 different treatments, and they were all just single-arm trials, none of them were randomized, and then they just looked at the median survival of those 4 groups, and they did a statistical test combining information across the studies to test whether any of them were significantly different from each other. And basically, the p-value was big, and you couldn't reject the null hypothesis that any of the treatments was better than any of the others. So what he tried to put in the paper is, therefore, we've proven that all of these treatments are equivalent to each other, and it doesn't matter which one you give. And so I was like, well, no, wait a minute. Maybe we shouldn't say things that way. And so here he's going to write in a medical paper this very flawed conclusion, when really the treatments are different, and they might be working for different subsets of patients. They may have different toxicities. And honestly, just if you have a larger sample size, maybe one is better than the other. So I had to dial it back. So I'll have to a lot of times look at how they're interpreting things, make sure it's careful. Another thing that I see a lot is over-interpreting descriptive results that have no rigorous statistical support. So they do a cluster analysis and say, hey, look, this stuff clusters together, let's interpret it. Or they do a whole bunch of multiple tests and they just report, oh, look, these things look like they're the most different. Well, yeah, they may be the most different, but statistically, can you really be sure that in the population that difference is real, that it's more than you would expect by random chance? Okay, and make sure that the strength of the conclusion is supported by the analysis performed. Okay, again, this also gets at these multiple testing issues. And so one thing that separates us from other quantitative scientists is our ability to understand variability. Because there are many quantitative scientists out there that are analyzing data, that are doing quantitative things. Computer scientists, some of them are called data miners, signal processors, mathematicians. But what really sets us apart from them is we are the best experts at understanding variability, understanding how to weigh evidence based on probabilities. And that's extremely important in this big data world, for us to carefully assess whether the patterns we see in the data are likely to be real, or might they be just spurious. And we need further study to really validate them before we can consider it a conclusion we draw. So all this stuff, not even the analysis, but just carefully interpreting the results is very important. And so we need to make sure in the write-ups that our collaborators write things accurately, conveying the degree of evidence their study has. Okay, so third area where we can make a deep contribution is experimental design. And this is one of those things that seems like it should be obvious, that scientists should know how to design an experiment to answer questions carefully. But it's amazing how many times even people who are otherwise good scientists will design their study in an extremely flawed way that prevents them from ever learning anything from their data. It could be disastrous many times. And my first experience from that was when I was a grad student. We had a consulting class where we had graduate students from other departments around A&M come and work with me. And one guy came and talked with me, and he had spent 2.5 years collecting his data in the field, and he was now ready to analyze it. And the way he collected it was his factor of interest was completely confounded with another factor. So he literally could not address the question that was in his dissertation with that data. After 2.5 years of collecting the data, he learned this. So it was kind of bad news for him. And if he would have come for help on experimental design up front, he could have prevented that problem. Now, one thing I'm going to do is talk about a case study. Well, Well, before I do, I'll make this general point. The reason why experimental design is so important is the data we get is only as good as the design and the sampling that was used to obtain it. And so if you have data that's from a confounded design, or that was sampled in some biased fashion, or even was pre-processed wrong, the data that you're analyzing, you could use the best statistical methods in the world or come up with some great new method and analyze it. It's not going to help if those experimental design issues are not addressed, and it's garbage in, garbage out. And so if you have a garbage design, it doesn't matter what you do to the data, you're going to end up with misleading results many times. And so that's the idea of why experimental design is important. So I'm going to talk about this case study here that I was involved in that sort of shows how much impact this type of thing could have. And so there was this paper published in The Lancet in 2002 saying that they could detect ovarian cancer early with a blood test using proteomic data. And so what they did was they collected proteomic data on blood serum samples from 100 women with ovarian cancer, 100 normal controls, 16 women with benign disease, and they split the data set in half, the cancers and controls, 50 normal, 50 control, and they trained a statistical algorithm to distinguish between cancer and normal. And then they tested the algorithm on the remaining samples as well as on the benign disease to see what they found. Well, what they found is all 50 of the cancers were correctly classified as cancer. 47 out of the 50 normals were correctly classified as normals, and there were 3 out of 50 false positives. Then all 16 benign disease were classified as not being cancer, but also not being normal, being something else. So that's why they got this big Lancet paper, high profile stuff, remarkable. The reason this was so exciting is ovarian cancer is typically not detected until it's very late. It's very hard to screen for ovarian cancer. And by the time it's detected, it's so late, it's hard to treat. So many women die of ovarian cancer because it's detected so late. So if we could detect it early with a simple blood test, it could potentially almost be able to eliminate the disease and definitely save a lot of women's lives. So this was very exciting. And I was on the front page of the New York Times. There was all of this media attention on this study. And they actually started a company and a product called OvaCheck, that this classifier that they developed, they were going to start developing it and market it and sell it to women to tell them whether they had cancer or not based on this blood test. So very exciting. So as a result of this excitement, of course, being at MD Anderson, all of our collaborators in all their different cancer areas said, hey, well, we want to do the same thing. We want to measure this proteomic stuff in the blood, and we want to detect our cancer early. This is amazing. And so we had to figure out how to analyze this data and design studies so our collaborators could try and do this. And so two of my colleagues in bioinformatics, Keith Baggerly and Kevin Coombs, and I then got involved trying to learn about this proteomics data. And in doing that, we downloaded their data and tried to reanalyze it so we could become familiar with it. But then we found a lot of major issues here. So one thing we did was we downloaded their data. And one thing I want to say is this proteomic data gives data like this, a proteomic spectrum, where you can think of the x-axis as being molecular mass, you can think of the y-axis as abundance, and you can see these peaks as basically proteins in your sample. And that's very, very rough description of it that Dr. Dabney would beat me up for because he's done a lot of work in this area, but it gives you kind of a big picture idea for what this data represents. So here's a heat map where each row is one of these functions where instead of the y-axis showing the intensity, the color shows the intensity. So the hotter colors correspond to the taller peaks. So if you just look at this data across all the 100 cancers, the 100 normals, and the benign disease, you don't really see a big signal with your eye, but what jumps out at you is this. The benign disease are very different from the cancer and normal. And the cancer and normal are actually more similar to each other than the benign disease. So that's kind of curious. That's not what we would expect. But then they posted another data set that was the same samples run on a different proteomic chip that subsets, because what happens is this chip binds with a certain subset of the proteins. So they reportedly used one chip for this analysis, then they reran it on a different chip type. So here's the heat map that we plotted of their data here. Now you don't see quite the big difference in the benign disease from the others that you saw before. But if you plot these heat maps against each other, we see a little problem. So can anyone see what they did? Just based on this picture, can you see what they did in the first study? See if anyone's raising their hand. See if anyone's figured it out. So does anyone have their hand raised? So what do you think happened here?
It looks like they shifted it to the left.
Yep. And I told you that this data set was run on one chip type that subsets one subset of proteins. These were run on a different chip type that subsets a different set of protein. So what do you think happened here? So do you think that they actually used the first chip type for these samples? No, they actually very obviously used this chip type for the benign samples in the first study. So when they were able to say, yes, we're able to classify these as not cancer or normal, well, yes, of course they could. But what they were really saying is, we can tell if you subset a totally different set of proteins, you get totally different data. Well, they didn't realize this. But we kind of realized this by just plotting their data and looking at it. So that's kind of a problem. So that's sort of shocking. But that's okay. Maybe the cancer normal difference is still okay. That's the most important anyway. So they did a follow-up study with another independent set of cancers and normals. And here's a heat map of their similarity matrix among the cancers and the normals. And so the blue color shows they're very similar to each other. So we can see the cancers are very similar to each other. The normals are very similar to each other, but they're different from each other. And this is the kind of heat map you want to see if you really can separate fit groups from each other. So this is exciting. So they say, okay, we validated our results. But they chose a different set of proteins on this new data set. So what we did is we went back and took their algorithm from the first data set and applied it to the second data set. This is the heat map. So can you see the normals and cancer separated from each other? Not at all. It doesn't. So basically, if you took the proteins that separated here and applied it to the first data set, they don't work. If you take the ones from the first data set to the second data set, they don't work. So that didn't seem to us that it's reproducible. So we actually published a paper showing that, because they were actually moving forward with this and starting to market this product, and lots of other things were going on based on it. So what went on? Well, here was another study where this is an actual figure from their paper, where they were showing here that this was day 1 samples. These were the samples run on day 2. These were the samples run on day 3. And this is showing a quality control measure. And the reason they showed this in their paper is they showed that something went wrong with the instrument on day 3. So they did some sort of threshold on the quality control, and they threw away those samples and only kept the ones above the line. But what's interesting is they also plotted the cases and the controls, that if you plot those together, this is all of the cases, or the controls. These are all of the cases. And if you overlay those, here are all of the controls, here are all of the cases. So you can see that they ran all of their controls on day 1 and just a tiny bit on day 2. They ran all of their cancers on the rest of day 2 and on day 3. And they acknowledged that on day 3, something went wrong with the instrument. So should we be surprised that in the data, cancer is different from normal? Probably not. And is that difference due to cancer biology, or is it due to possible confounding factors from the instrumentation? Okay, well, you can obviously see there's a major problem here. And it turns out that the reason they got such dramatic results is they confounded the run order with case control status. And that's also why their signal would not reproduce. So it was kind of scary, because they were about to put this on the market in 2004. And it was not thought to be under the jurisdiction of the FDA, because it was considered a homebrew laboratory test. And no further studies published showed the test worked. So we published our paper, and Keith Baggerly went and he's become like the master of reanalyzing and what we call forensic biostatistics. and this is the first case, and he's done other ones like this where he's really made a huge impact. But here, our paper was published. Then the FDA saw our paper and actually had talked to Keith Baggerly, and then they sent a letter to this company saying, you can't market this. We need to double check it. Then they said, you can't market it. It requires FDA approval. And they gave them parameters of other studies they had to do to validate it. Of course, they couldn't validate it because their pie in the sky results were because of a flawed experimental design that gave them false positives. So they actually were never able to get it validated. So really, honestly, that work that we did is really what uncovered this. And so as statisticians, we were able to really intervene and help an ineffective test from being given to women. So that's kind of a dramatic story, but the basic story there is they never plotted their data and they didn't think carefully about experimental design, and that led to the whole fiasco. Okay, so a last point I want to make is, so how long does class go here? I want to leave some more time for questions.
You got about 20 minutes.
20 minutes. Okay, so I'm going to try and finish in about 10 minutes at the most to give time for more questions. So another way we can make deeper contributions is helping to refine their scientific thinking. And this is something that I find interesting. And when I'm helping them plan studies, of course, I help them with their experimental design to avoid disasters like happened in that ovarian cancer case. But a lot of times I find my greatest contribution is not the analysis plan or even the design, but helping them refine their thinking. Because many times they know what they want to study, but being able to formulate those into clearly testable hypotheses and design their study to really answer that hypothesis and help them to think about what the data they can collect and what questions they can and can't answer. And many times then, once we talk about the data, I say, well, if you collect that data, you can also address this question. Is that interesting scientifically? And many times they'll say, oh yeah, that's very interesting. And so sometimes having that discussion about the data and the design and all of that, at the end of the meeting, they actually can understand their scientific questions more clearly because of the quantitative input that I provided. And so sometimes we can help them even identify new hypotheses or find new ways to integrate the data that they have to learn things. And so that's where I find that I can make a deeper contribution as a collaborating scientist, that it's not even just the technical stuff that I'm helping them with, but just because of my quantitative mindset that I come to the problem with, I'll help them to think more clearly and design their study and design their research better. So this process requires that we try and understand the science and learn to communicate, which is kind of hard work, but if you can get good at that, you'll be a star as a statistician, because that skill is one of the most important, being able to communicate with others and learn. So that's a good thing for you to work on no matter what you go into, whether it's industry, academia, whatever. So now I'm going to finish with just kind of a case study of some really exciting things I'm working in now, where I'm getting to be involved more deeper in the main science and not just in the technical side. So here this is just an illustration of precision medicine, which is the big thing in medical research right now, what we're trying to do. Because classical traditional medicine treats everybody with a disease the same with what they consider the best standard therapy. So here, this for colorectal cancer, this represents men and women with colorectal cancers, and the colors represent sort of different molecular characteristics of their cancer. So the old way was we just treated them all the same. Some of them did well, some didn't. We just tried to treat as many well as we wanted. So sort of our goal is to do precision medicine where each patient gets an individualized treatment based on their particular disease. That's sort of the goal, but statistically it's hard to do because if everybody's disease is individually, then we have a sample of size 1 for every disease, and we can't know anything about that person's disease from other people. So it's kind of tricky. How can we find some middle ground between this and this? Well, one way to do it is to identify subtypes of the disease that are maybe they're individually slightly different, but they're more similar to each other. And then if we can find subtypes, especially using the molecular biology, then we can find specific treatments for those subtypes that are a more targeted strategy than this, but we have at least moderate sample sizes to study and learn, as opposed to this. So that's kind of the idea. So if we could find these subgroups, we can use them prognostically. So prognostically means find subgroups that are likely to have poor outcome, and then those patients we can treat more aggressively because we know that they're probably going to have a poorer outcome, and it's worth trying a more aggressive treatment. We can also try and use these predictively, where we can find subgroups of patients that respond to a given known therapy, because there's different treatments that might work 10 or 20% of the time. But if we can identify which 10 or 20%, we can do a much smarter treatment strategy rather than just trial and error. And another exciting thing we can do is if we find these subtypes where there's really different molecular biology, we can study that biology and find a new drug target for that specific subtype. And because we now have found a subtype that is more homogeneous, we might have greater sensitivity to discover the new drugs, where if we would have studied things, tried to find the drug here, if you have a drug that only works for this subtype that's 10% of the patients, if you try it here, you might just think it doesn't work. But if you have the refined subset, then you may be able to have more power to find a specific treatment. So here, some of us at MD Anderson joined this consortium whose goal was to find consensus subtypes of colon cancer. And so in this consortium, we combined information across 18 different studies of gene expression, over 4,000 patients' data, and 6 previous subtyping systems to find consensus subtypes. And then we published this a year and a half ago in Nature Medicine, that there were 4 consensus subtypes with these proportions. And so this is just sort of a cluster plot. Each dot is a sample, and it just shows how the patients sort of cluster into these groups. Not perfectly, but you can see these distinct subtypes. And when we look at the molecular biology in these subtypes, each of these subtypes has very specific biology going on in that subtype that is not in the other subtypes. So this had us very excited, and really the whole colon cancer research community is very excited about this and trying to study these subtypes and apply this to new learning for how we can treat colon cancer. So here is a heat map that just shows the gene expression data for a bunch of patients in the training data and in a validation data set. You can see this very large data set. So each column is a patient, and then each row is one of the genes that we chose as part of the classifier we build to classify patients into these subtypes. But what I want you to note is, so red is high and green is low expression. But one thing I want you to note is in this independent validation data and training data, look at how similar the heat maps are in distinguishing, finding genes that are really have different gene expression across these subtypes. So this is the type of thing we want to see when we have a reproducible biological signal. Okay, so we're very excited, and this data set is large and diverse enough to hopefully find the true signal in the data. Okay, and so the gene expression is only a part of the story. So remember in the beginning, earlier in the talk, I showed there's all those different types of multi-platform genomics data we have that look at DNA, that look at methylation, look at proteins. Well, we have all of those measurements in one data set that the NIH collected called TCGA that has 250 samples. Then for 220 MD Anderson patients, we have all those same measurements. So what we're trying to do right now is to deeply characterize the molecular biology of these subtypes. And we have these 2 data sets, so we can learn on one and validate the signal on the other. And so again, what we hope to do here is to find specific precision therapy strategies for colon cancer to identify the patients that for more aggressive treatment, to find which patients should get which treatments, and we're also trying to find new drugs, new drug targets, and even develop new drugs that can target the biology in these subtypes. And at MD Anderson, we have one department that really does its own drug development. So we're working with them to try and identify some targets that could lead to new treatments for some of these subtypes. And so the key to this learning is integrative modeling of all of this data, which is a statistical problem that's one of the hottest open problems, I think, right now in biostatistics. And so we're involved in some methods research to also discover some of this structure. But I'm very excited because I'm hoping that some of these new methods can identify new medical and biological truth that could be used to treat patients in a different way. Okay, so in conclusion, so what I love about statistics, I'm so excited I found this field. I think it's the best, coolest thing. It's great for me, for what I enjoy. I get to use my analytical skills to solve real world problems. I get to play in everyone else's sandbox. I get to interact with many different people all over the world and learn lots of things. And also there's great job opportunities out there. They pay well, good quality of life, very interesting. And what I'm most excited about is great opportunities for impact, because that's kind of really my goal in life in general, as well as in work, is I want to make an impact wherever I am, whatever I do. And I think being a statistician, you have a chance to make such an impact in not just biomedical research, but any societal problem that involves data. We sort of have a skill set that can really help discover the truth and get the information out there and help make an impact. And also I get to mentor and teach others. It's a very collaborative field, so I also enjoy that. And that's one thing I find is that in many ways, the biggest impact we make is our interactions with others, in our families, with our colleagues, with other people. So it's really a field where I get to interact with a lot of people and hopefully make many different types of impacts. So I'll stop there, and if we have a couple minutes, I can take any questions on either the substantive stuff or the big picture stuff or professional stuff, whatever you guys want to ask.
Let's thank Dr. Morris. You had your hand up. Go ahead.
Okay, so out of the past projects you've worked on, what is your favorite project so far?
What's really exciting to me is my favorite project keeps becoming the next one I'm on, which is what you want to have happen. But right now, like the last one I hinted on, this colon cancer subtypes, I'm super excited about. And what's my favorite about that is not necessarily the statistics. There are some statistical problems, but it's just that I feel like we're on the cusp of kind of redefining colon cancer and the way it's treated, and the opportunity to be a part of that and maybe even make a strong impact is so energizing. So that's really exciting to me. But then statistically, like some of the methods, I didn't talk anything about the methods I've worked on functional data analysis, but that stuff that I've been working on really since my dissertation, I started working on that area. I've continually worked on that, and my goal has been really developing a set of flexible methods that could apply to a lot of these types of data and allow investigators to analyze more of the raw data in its complexity and not just simplify the data and extract summaries. And so that whole work I'm still very passionate about, and it's kind of coming together where I'm developing it into a software package and applying it to some exciting new areas. So methodologically, that's very exciting to me as well. And some of that stuff is just enjoyable to me because the mathematics and the statistics behind it are just really cool, even outside of the practical impact. So I enjoy that for its own sake as well.
Okay, go ahead.
Okay, so I'm currently studying statistics here. I'm a junior, and I'm under the bioinformatics track. I want to pursue cancer research as well. And
Great.
I already have my mindset. I'm going to grad school for that. But I'm just wondering, what advice do you have for me?
So I guess the advice I would have, one advice I would have is just learn everything you can. Just work really hard, make the use of the time you have, make use of the resources you have, of faculty, other people you can learn from, and just try and soak it all in. Specifically in bioinformatics, I would encourage you to learn as much molecular biology as you can, and also develop computing tools and programming tools to deal with the big data as much as you can. Because one thing about the bioinformatics and this multi-platform genomics is there's big data management issues and such like that. And I know I'm not very good at that. I'm lucky that I have a lot of collaborators and people here that are good at that. So I can, still work in this area with them, helping manage the data. But I would feel much more free if I understood better how, some of the programming languages that are used to deal with that big data. So that would also help you out. But I would also, encourage you to figure out, even bioinformatics is a very big field. So identify what aspect of bioinformatics interests you the most and try and really identify, a program and an advisor to work with where you can, do the type of thing that you're interested in. But also I would recommend keep reading, keep up with the scientific literature, because one thing about this field is every 2 or 3 years there's new things, that new measurement devices and new things they're able to do that changes everything. And so keep up with sort of the latest and make that a habit, because it's a very quickly changing field. But that's also what's very exciting, because there's stuff we can do now that 2 or 3 years ago you couldn't dream of, and that just continues. Very exciting.
Go ahead.
Speaking of new technologies, have you ever had the opportunity to work with CRISPR in your research? Or?
Yeah, it's funny because I was almost going to start talking about CRISPR as the example I was thinking about, but I didn't want to start going on about that. I haven't worked with it directly, but we've hired a new faculty, Trevor Hart, who worked at the Broad, and he has first author Nature papers on CRISPR. He's one of the leading guys on CRISPR, and so that he's brought a lot of expertise on CRISPR to MD Anderson, and now we're starting to incorporate it. And even in our colon cancer group, we're trying to get him involved so that we can use CRISPR in sort of studying these subtypes and identifying the targets and things like that we want to do. So I haven't directly worked on it yet, but I've talked with him about it, and it's taking off. And I know that in some places that has just become a whole field in itself. They're just doing it all over the place, but it's very exciting. And for those of you that don't know what CRISPR is, it's a very exciting new technology where it's actually basically gene editing, where they can basically go in and change the letters of the nucleotides in the gene sequence to change the genetic sequence. So it's really very much like the stuff of science fiction that people would make movies about, but it's actually possible now, and they're developing it and using it. And so it's, because you can go in and change one thing and then see what it affects biologically. It's mostly done on cell lines, just cell lines, not in vivo systems. But I think they're trying it in animal systems as well. Obviously, trying it in human studies would, be a bit off. But yeah, very exciting.
Any more? Go ahead.
We hear a lot about statistics and how it relates to nutrition and biological sciences and stuff like that, but do you know what, if any, kind of role statisticians can play in the harder sciences like physics or chemistry?
Oh yeah, definitely. There's a lot of impact. I think part of the reason why biomedical research is talked about the most is because there's so much funding and so much research going on. It's so really in the last 30, 40 years, you look at how medical research has taken off. Just look at the number of people working in that research. That's why it's so big. But honestly, the other physical sciences have great needs for statistics too, and some of them even need more advanced statistics. One area, think of astronomy. Astronomy is tons of statistics in there. And quantum physics, it is statistics. People in physics, a lot of times what they're learning is really, statistical mathematics undergirding physics. But even in engineering, there's a lot of statistics there. And manufacturing is not as big as it was, a few decades ago, but quality control is a huge part that has pushed manufacturing ahead in the last 50 years, and statistics made a great contribution there. And in signal processing, in terms of all different types of signal processing within electrical engineering is basically statistics as well. So it's really all over the place. And even one thing that I don't know if any of you guys like love the West Coast and love the glamour of working for a company like Google or Facebook or any of those companies, but those companies love hiring statisticians, and there's many of the things that they're doing, developing new technology like self-driving cars, all those things. There's statisticians working on all those projects. Like I know, some statisticians that are working in those companies doing really exciting things, and that's a career path you can take. They might even hire undergraduates in some of those companies, but then also if you're really, really interested in that, there are certain universities where their PhD programs have great pipelines where a lot of their students get hired into those companies. So that's another area. So really it's all over the place where statistics makes an impact.
Anybody else? Okay, well, I think that's all the questions now, Jeff. So this is Tom Morley. I took over for Alan.
Right.
And so I enjoyed hearing you, and I'd like to thank you a lot for all the interesting ideas that you gave our students.
Thank you. Appreciate it.
Okay, have a good day.
Okay, you too. Thanks. Thanks. Bye-bye.
Okay.